Good day, everyone. In this presentation, I am going to discuss what is all about the active transport, how does it works, and what are the different types of active transport. Okay, so active transport is the total opposite of passive transport. Okay, so the movement of molecules from the membrane or across the cell membrane is from the region of low concentration to a region of high concentration against the concentration gradient. And lastly, active transport requires the input or the use of energy known as the ATP or adenosine triphosphate. Okay, so moving on, we have two types of active transport, namely the cell membrane pump and the vesicle transport. Okay, so first, let's discuss what is all about the cell membrane pump. Okay, so this membrane pump involves the carrier proteins to move the materials against the concentration gradient. Of course, with the input or use of energy. Okay, so one example of cell membrane pump is we have the sodium-potassium pump. Okay, so this sodium-potassium pump is a pump that moves the sodium ion out of the cell and the potassium ions into the cell. Okay, so we have four primary steps in sodium potassium pump. Okay, so first is three sodium ions will bind with the carrier protein in the cell. And then the carrier protein will receive phosphate group from the ATP. Okay, so when the ATP loses a phosphate group, energy is then released. Okay, next is and with the aid of this energy from the ATP, the sodium ions are now released to the outside of the membrane. And then the new shape of the channel protein will allow the two potassium ion to bind. Okay, so again, there is a release of phosphate from the ATP to release energy. Okay, so again, when we have the energy, the channel will release the potassium into the inside of the cell. Okay, so those are the four simple steps of sodium potassium pump. Okay, so what is the importance of this sodium and potassium pump? Okay, so getting the balance of sodium and potassium pump or sodium and potassium in our diet is important for good health because imbalances of the sodium and potassium intake can increase the risk of high blood, heart disease, diabetes, and other disorder. Okay, so moving on, for the second type of active transport, we have here the vesicle transport. Okay, so some molecules are too large to pass through the plasma membrane regardless of their concentration inside and outside of the cell. Okay, so but through the help of this vesicle transport, these large molecules can cross the plasma membrane. Of course, the process will require the direct use of ATP to fuel the transport of this large particle. And we have two types of vesicle transport, namely the endocytosis and the exocytosis. Okay, so when we say endocytosis, it is the process by which a cell membrane invaginates and form a pocket around the cluster of molecules. This pocket pinches off and form vesicle or vacuole that will transport the molecules into the inside or into the cytoplasm of the cell. Okay, so we have three types of endocytosis. We have the phagocytosis, the pinocytosis, and lastly, we have the receptor-mediated endocytosis. Okay, so in phagocytosis, the cell will ingest external fluid, macromolecules, and large particles including the uh, old cell part, old tissues, viruses, and bacteria. Okay, so phagocytosis is also known as cellular or cell eating. Okay, so in preparation for the phagocytosis, a portion of the inward-facing surface of the plasma membrane becomes coated with a protein called clathrin. Okay, so which stabilizes this section of the cell membrane. In addition to that, clathrin is a protein that plays a role in the in the formation of coated vesicle. Okay, so the coated portion of the membrane then extends from the body of the cell and surrounds the particle, and then eventually closing it. Okay, so once the vesicle containing the particles is enclosed within the cell, the clathrin will disengages from the membrane and the vesicle will merge with the lysosome for the breakdown of material. Okay, so kindly take note that in higher organisms such as the animals and of course as humans, okay, so phagocytosis is mostly employed by immune cells for defensive purposes of engulfing dead tissues, dead cells, foreign invaders such as the bacteria and viruses. Okay, so in some lower organisms or animals such as the amoeba and sponges, phagocytosis means feeding. Okay, so we have here a diagram. First is we have first is ayan. So the plasma membrane will entraps the food particles. Then a food vacuole or a vesicle will form within the cell that contains the food particle. Next is the fusion of lysosomes with with the food vacuole. And then the enzymes from the lysosomes will digest the food particles. Okay, so when the food particles are digested, the nutrients will absorb by the cell and the waste will leave the cell through exocytosis. So this process is for the amoeba and sponges only. Okay, so in addition to that, phagocytosis also involved in the formation of pseudopodia or false feet for the intake of molecules in lower organisms. But take note that the speed of the phagocytosis varies with the size of food particle. So we have here an amoeba taking or digesting food under the microscope. Okay, next is we have an image here of a Staphylococcus aureus and a neutrophils. Okay, so Staphylococcus aureus is a bacteria. Okay, so this Staphylococcus aureus is a type of gram-negative bacteria which is a leading cause of skin and tissue infection. And for the blue one, we have here the neutrophils, which is a type of white blood cells that engulfing the bacteria. Of course, in the process of phagocytosis. Okay, so for the second type of endocytosis, we have the pinocytosis. Okay, so it is the intake of liquid and small particles, usually ions, amino acids, sugar, insulin, and lipoprotein. Okay, so unlike phagocytosis, pinocytosis is a routine or active routine of most cells, wherein pinocytosis is employed for the intake of nutrients. Okay, so one good example of pinocytosis is the absorption of nutrients in the small intestine. Okay, so the small intestine, as you can see here, it is made up of villi. Okay, so this villi are tiny finger-like projections that line the entire length of the small intestine. And these villi Ayan. It is made up or composed of microscopic cellular membrane of microvilli. Okay, so this microvilli aids for more absorption of nutrients such as the fats and vitamins in the process of pinocytosis. Okay, so for the last type of endocytosis, so we have here the receptor mediate endocytosis. Okay, so in the receptor mediate endocytosis, it is a targeted variation of endocytosis. Okay, so unlike phagocytosis and pinocytosis, there is a protein receptor in this type of endocytosis. Okay, so these protein receptors will bind with specific molecules to capture will bind with molecules to capture specific molecules such as the hormones and proteins and ions. Okay, so the clathrin will aid for the formation of coated pit around the receptor and the molecules. Okay, so although receptor-mediated endocytosis is designed to bring specific substances that are normally found in the extracellular fluid into the cell, other substances may still gain entry into the cell at the same site, such as the flu viruses and cholera toxins, especially when they cross-react with the normal receptor binding, and then they will gain entry into the cell. Okay, so exocytosis is much like the endocytosis in reverse, wherein materials to be exported is packaged into the vesicle inside the cell. Okay, so this vesicle containing the substances move through the cytoplasm to the cell membrane, and then the vesicle membrane fuses with the cell membrane, and the substance is released outside the cell. Example are the secretion of proteins, enzymes, and antibodies needed needed by the cell and the tissues. Okay, so when glucose concentration are low, we have here the glucagon hormones that are secreted from the islets alpha of the cell of the pancreas. Okay, so these glucagon hormones will signal the liver to convert the glycogen or the stored glycogen into glucose. And then this glucose is then released to the blood, causing the blood glucose rise to increase. Okay, so it is a example of a endocytosis and exocytosis, wherein the liver will produce glucose and the blood vessels will receive or intake this glucose in the process of pinocytosis. Okay, so another is that the pancreas also secrete digestive enzymes such as the proteases, lipase, and amylases that are very necessary, especially in the process of digestion.